So when we left off protein synthesis, we'd made this chain of amino acids called a polypeptide. Great. That's just a chain. It's like a string of beads. How does that make a protein? How is that going to make my hair? How is that going to make the hemoglobin in my red blood cells? They're all very different shaped molecules, and you're telling me that they're just chains? No, not quite. Those chains undergo a few different processes to make them fold in different ways and therefore perform different functions. So what we call this is the hierarchy of proteins. Now, hierarchy just means this is the order. We start off down the bottom with a primary structure. So here is our primary structure. The primary structure is simply that chain. Now, it's the direct end product of translation. So go back to the protein synthesis, you make that polypeptide chain, that's what we're talking about. This is the direct product of protein synthesis. What it is it's a linear sequence of amino acids in that polypeptide chain, and it is unique to every protein. So there are 20 different amino acids. You can just imagine how many different combinations you can have. You don't even have to have different ones next to each other. You might even have a whole bunch of threonines right next to each other. It doesn't matter, but these are like the fingerprints of your proteins. They are what make each protein unique. Once you change the order of amino acids, you're going to change your protein. That's it. Think about it as this is your base language. If this changes, everything else above it changes. This is primary structure, the bottom level. The next level is what we call the secondary structure. So now we've got this long chain of amino acids. And what happens with that is a couple of different things. So one thing that can happen with this chain is that it can form a bit of a coil, kind of like DNA. This one's not going to work very well for me. I'm going to try and do a double over. Kind of like DNA, where it just kind of coils around itself like this. And that's what we call an alpha helix. Now you can have a look there. If you want to be a chemist nerd, you can see that there are hydrogen bonds that are kind of interacting between R groups. And that's what's giving it this coiled feature here. Now, the other way that they can bend, there's two different ways that they can fold in this secondary structure. This is so you fit more of them in, and that's they can be what we call a beta pleated sheet. So these are two different things. When we talk about secondary structure, we talk about them being either, this is really not working as an example, either a beta pleated sheet, which is where they're folding back on each other like an accordion, or an alpha helix. So that's what we call the secondary structure of a protein. So it's either folding or coiling of that original chain. Now, once you've done that folding or coiling of that chain, we're going to move forward to our tertiary structure. Now, this is when we start to get cool three-dimensional shapes. So those coils kind of come together and might fold on top of each other. There might be some beta pleated sheets next to some coils. You might start to get some other funny things happening. You can see this picture on the right over here has got all sorts of things. So you can see here there's an alpha helix. There's also a bit of a beta pleated sheet going on down here. It also just kind of twists around on itself. And each one of these three-dimensional shapes is caused by that primary structure. It's like your building blocks. You can't create something different. So when you change this base level, it's going to change every level above it. And that's why we call it a hierarchy. So we've got a combination of alpha helices and beta pleated sheets. It occurs because there are different types of bonds. You can go into the types of bonds if you wish. There are, for chemists, we've got covalent bonds, ionic bonds, and hydrogen bonds. And when we talk about those three types of bonds, the covalent are the strongest, followed by the ionic bonds, and then the hydrogen bonds, because technically they're just an intermolecular force. The other two are a bit more of an interaction between the atoms. Sorry, that was a digression down the chemistry route. This is critical to protein function. If the tertiary structure changes, the protein most likely won't work anymore. Yeah. And what changes this is a change in the primary structure. So if you get a change in that sequence of amino acids, it's going to change your tertiary structure. And when that happens, protein might not work the way you want it to anymore. Now, some proteins also have what we call a quaternary structure. Now, this is the level above tertiary, and all it means is when you get more than one chain. 2 or more polypeptide chains, they've gone through primary structure, which is your chain, they've been alpha helicized and beta pleated sheets in secondary structure, and in tertiary structure, they've folded around on themselves. So you've got this tertiary structure protein, and then you've also got another tertiary structure protein, and sometimes they join together. And if they join together, that's what we call a quaternary protein. More than one polypeptide chain joining together. Hemoglobin is a really good example of this. Hemoglobin is made up of 4 polypeptide chains or 4 tertiary structures bound together. And so some proteins don't work unless they have multiple chains to them. You can see how complex proteins can become quite quickly from this. Now, what I'm going to say is I'm going to send you a way to actually complete this sheet. Hopefully it's printed for you. If not, it should be available on the MyCGGS LMS for you. And that actually sums up the end of protein hierarchy.